behind methodological issues. So the expected changes from placebo are too high. For example, if you remember, sometimes, well, 25% may be reasonable, but sometimes 40% and 50%. Almost this is expecting to negative result unless you hit the home run. So because of the budget issues, you go higher expectations, so you cannot get small positive result. Also, disease at enrollment was already to progress. This is another issue. Already when you make diagnosis, Dr. Sober's studies, motor neurons are so much lost. So how we can do earlier, that's another issue. And also disease at enrollment, yes, I'm sorry. And two primary endpoints caused confounding result. So some study used two endpoints such as survival and also pulmonary function test, and that was an improvement test, and very difficult to interpret those two results. Also, study period was too short, which was cited, but probably you can interpret other studies also. Study period was too short. This was not our interpretation. This was again repeatedly trial investigators' comments. So how about disease diversity, heterogeneity is another issue, and again upland orange problem, and then imbalance of enrolled patient, and that was rather common. I mean, simple problem that could happen after enrollment, and requiring different phase 2 studies, and that was probably sometimes we want to go too fast, move to phase 3 with small phase 2 studies, turned out to be more nightmare after phase 3 studies. Patient population different from phase 2 studies. Again, when you get phase 2 result, you want to do something better phase 3 studies. So not necessarily repeating phase 2, but more modified phase 3 turns out to be sometimes against the result. And rapid enrollment is always praised with good studies, but not necessarily rapid enrollment, moving some strange patients enrolled because we need to enroll fast. So off-label medication is easily available for anyone, which was often investigators initiated clinical trials such as gabapentin and all those medications available. Is very difficult to conduct placebo or controlled clinical trials. So disease-modifying clinical trials has full of issues, but symptomatic clinical trials are a bit different because now this is not for you to read. Sorry, this is copied for paper. For example, spasticity, respiratory failure, pseudobulbar affect, PBA, and also bronchial secretions, bone fracture, and muscle weakness, fatigue, and sialorrhea, and physical activity and coping, and muscle weakness, weight loss. Those are really try to improve symptoms. It turns out to be those symptomatic treatment is somewhat loose because not very strict placebo-controlled clinical trials, and also loss of patient attrition rate is relatively high, and so there's a clearly symptomatic clinical trials has more problem in terms of method and design and so forth. Yet symptomatic clinical trials turns out to be more high yield because now we have approved medication such as pseudobulbar affect, as you know, combination of quinidine and dextromethorphan, and TIV is now widely used and used through guidelines, and sialorrhea, Botox are widely used based on those papers. And so if you look at, there are potential medications, for example, muscle weakness. Now another study will be performed, I hope, and Tecentriq is very hopeful medications and so forth. So if disease-modifying clinical trials are difficult, symptomatic clinical trials are also very important to improve quality of life. So suggestion for future randomized clinical trials and expand research to find the cause and pathogenesis of ALS. And always we think randomized clinical trials can offer the best opportunity to study those pathogens and cause. Yes, indeed, clinical trials are only one purpose, to test the medication and bring medication to the patient. But at the same time, this is the greatest opportunity to find the underlying disease cause. Investigate the drug target in human patient and was found in preclinical trials. And the problem is, many medications, we do not know in human what medication get to the target. Not physiologically target, but working in a target, we don't know. Also incorporate biomarker studies such as creatinine. And again, biomarkers are coming to reality, so I think we need to incorporate biomarkers. Preclinical studies models, and Albert published with the other experts, followed the guidelines published in 2010, and I think ideally incorporate human randomized clinical trial methodologies. Now, really important to do best available preclinical studies in animals, and that's essentially like human studies. So confirm and duplicate the studies at independent laboratories, very important, and you cannot rely on one studies. Consider investigating the candidate drug alone and with Riluzole. Again, Riluzole still remains to be only one medication. We have to do testing with Riluzole in preclinical stage. And through transaction from basic science to clinical trials and vice versa. And again, I'll tell you, basic science finished their job when they find the medication. Clinical trials pick up that medication and then try. That's complete, almost a different world. And I think we have to cross real translation from basic science to clinical trials, and clinical trials back to basic science. That's extremely important. Also evaluate the validity of preclinical studies by evidence-based and medicine analysis and an advisory board of scientists. I'm going to show you this. And then develop new disease models for ALS. This is again another essential directions. We need multiple good disease models. So continue doing some suggestions. Include other disciplines in randomized control trials. Clinical pharmacology must be in a randomized control trial. We don't know any clinical trials pharmacologist is a major player in the studies. And also pharmacologist has to be in the preclinical studies as well, in animal studies. And translation basic science is assisted to identifying drug target in randomized clinical trials. So to improve the design and methodology of randomized controlled trials, genetic and screening tests for cognitive impairment at enrollment is another thing already we discussed. And consider significant longer study durations. We know disease durations changes in the 1990s when Riluzole was published or studied, and now disease clearly slower, patient lives longer. So therefore, just a regular 18 months trials for survival may not be long enough. We need to find out. So encourage the development and application of innovative phase 2 studies. So phase 2 studies are very important. Consider more phase 2 studies before pivotal investigations. And well, we cannot rush to pivotal studies. And consider specified a priori post hoc analysis to find potential response group and reasons for study failures. Well, we may not have failures, but at the moment, so many clinical trials has has failed, that means likely to have more failures, to be honest with you. So therefore, we should make pre-specified post hoc analysis. So we should find out why it went wrong. And so I think it's post hoc analysis was highly criticized by, say, data fudging and so forth. But if you make trials, when you make trial design, I think that's a different story. Additional studies on natural history and phenotype variation is again very important. We need to discuss more. So consider more randomized controlled trials for pharmacological and non-pharmacological symptomatic treatment, as we already discussed. And then finally, plan a meeting with international clinical trials with experts in other disciplines such as regulatory agencies, funding agencies, pharmaceutical companies, and patient advocacy group to discuss future randomized controlled trials in MS and upgrade or update the second early house workshop product, which was guideline published in 1994, first time, second one 1999. So this is the graph, and just I'd like to reiterate once again, because clinical trials, I mean preclinical studies on the left side and clinical studies on the right side, and then the translational scientists, the base scientists find the medication and give it to clinical scientists, and then there is a big disruptions. There is no continuations. I think basic science has continued, but at the same time, pharmacologists as a biostatistician work with the clinical trials. Pharmacologists should work with animal scientists and also clinical scientists. I think it's more true translational research, not stopping at the beginning of clinical trials. That's probably important. So those are our recommendations. So not necessarily means Ben, Vincenzo, and myself recommendation may not be you agree, but I think we need to discuss those issues widely and plan for future clinical trials. And already we set early house 2016, not next year, but later after next year, and February 30 to February 6th, and in early house, and web conference will be available if funding doing well, and also evidence-based review of extensive issues involved with various clinical trials. So this is evidence-based. That means much more elaborate study. We have to start very early and starting from February to do evidence-based review, updating the old guidelines and building the new guidelines, circulate the new guidelines broadly and publish them online, and then in reputable neurology journals. And this is not the end of it. And disseminate and implement the guidelines for the future clinical trials, also responsibility of evidence-based medicines. So this is our future. Early House 2016, and now we have organized a committee. We have volunteers, and for example, Mike Bernata, Brooks, Brewing, and Conway, Sukovich, Dicky, Gilbitz, Katz, Rudolph, Miller, Mitsumoto, Robert Shaw, and Silani, all volunteers will organize and study with evidence-based medicine. And also we have the International Advisory Board. This is a planning stage, not completed, and all organizations such as ALSRG, which is maybe unfamiliar to European. This is ALS Research Group, American group, INCALS, NILS, WALS, WFN, all those are more larger organization to support this early house. And all stakeholders, as I mentioned, for clinical trials, including everyone, stakeholders, should be hopefully in face-to-face meeting or involved with website meeting to develop the new guidelines. we are seeking funding from various organizations, and some are already funded, and some still waiting, some are applying. So that's what our plan. So this is just a brief summary of my talk, and expecting some eggs and tomatoes.
Questions?
So all of the things that you've mentioned seem highly relevant, but the rate-limiting step, most people would probably agree, is failure to really appreciate the biological basis of the disease in enough detail to design drugs. So one could intuitively say, well, let's, as part of clinical trial criteria, establish a set of principles that will allow a drug to move from the preclinical to the clinical arena. And that would work very well if you had good models, if you had good understanding. It's a sort of circular problem. And, you know, if we establish a committee that says, well, you know, you can only take a drug in clinical trial that it fulfills, it works in this model, this model, this model, that might be good, but it might also limit our sort of imagination and our freedom to do trials. What do you think is the right approach?
I think guideline should not be limit clinical trials. So I think it's just we have discussed in chatting, and Massachusetts speed law, so the speed has to be reasonable. So kind of guidelines, I think it's not limit but encourage. And so I think we can point out what needs to be done. And for example, yes, clinical trial is the best opportunity to find the cause and pathogenesis. You cannot do everything in clinical trials, but you can do something, one thing, maybe biomarker, maybe target, whatever if you do clinical trials. Just I'm trying to say is not the limit, but I think it's encouraged to do more clinical trials. But yet something fundamental things should be discussed. I think it's this morning's discussion about the diagnostic criteria, and the staging. I think this clinical trial guideline will be very exciting meeting. As I don't think we can get easily agreement many aspects of this area, but I think somehow whatever we can agree very low level, that's what we have to find out. But those low level is high enough to lead clinical trials in better direction. That's what I'm not sure I'm answering your question.
Leon.
Yeah, very nice. I also agree with everything you said. Is it one thing that may miss is that so Biogen spent $100 million on a beautiful trial, I think. But so now it seems like that's the rule now, that you need at least $50 million to start a phase 3 trial. I know that companies are looking for those amounts of money. Do you think we as a ALS community also have a responsibility to look for ways to lower the costs of phase 3 trials, to make it more simple or more efficient, so that it's not. I mean, we've done, I know also in the UK, we've done lithium trials for under $1 million. I hope companies here don't listen. But I mean, it could be probably a little bit cheaper. So what do you think? What role could we play in that? Besides that, the salaries should be lower.
That's such an important point. I don't know. I have no answer, obviously, but I think we need to incorporate regulatory agencies, people, EMA and FDA has to come. I'm sure they don't say to limit the cost and so forth, but I think we as a researcher and in clinical trials to set some guideline. The importance of evidence-based medicine. I'm sure everyone knows about evidence-based medicine, but if evidence-based medicine recommendation comes, that is more persuasive, more forceful. Therefore, if we come up with those guidelines, the pharmaceutical company may need to pay serious attention. So if our guide, for example, if our guideline says cost, clinical trials cost has to be reasonable, has to be you know, to lower down so we can do more clinical trials, not the big phase 2, but phase 2, not the phase 3, excuse me, but more phase 2, which is clearly less costly. Why you have to do phase 3 to find the negative result? Maybe phase 2 is enough. Kind of things. Maybe we can make recommendations as guidelines, evidence-based. If hopefully, that's probably more persuasive to drug company. That's my guessing.
There's one question.
Yeah, I think when I look at this field, and I'm relatively new to this, by the way, I was trying to find out what failures in trials were, and until I read your paper, so thanks for that, because you already did all the work. What I think is very striking is that we are in a situation where big pharma is not very attentive to this field. And big pharma is traditionally very good at doing trials where we do as less as possible, because we need to abide to the guideline. And we don't want to find, and I've worked with the big pharma, with J&J, and we try to leave out whatever can raise a question. Now, when I look a lot at the ALS trials that I've seen so far in your review paper, in those trials we try to investigate a lot. And the reason is that this is a field that is driven by academia, where we try to find out new things, because we just heard this morning that we do not know a lot about the disease. So on the one side, we're trying to investigate a lot in trials, and on the other side, you know, we don't want to investigate too much to make sure that we have a positive trial. Now, when I was looking at these 18 compounds that have been tested in clinical trials, you know, the daring question is, you know, what compounds are in there that are actually effective if we would have investigated these drugs in a more simple trial? Now, that is the daring question that I want to put out here in this community. What if we would do the trial in a more simple way, leave out a lot of the things that we would like to learn, but focus first on the small effect that we want to establish, because we know, because the disease is heterogeneous, that we are looking for small effects. So if you're looking for small effects, you want to leave out a lot of questions and focus on the main question. Now, to do that, we need to maybe have as a community a more big pharma mindset, devise trials in a way focused on finding these small effects, and apart from that, design other types of trial where we try to learn what the disease is all about.
Right.
This is maybe a type of thinking that we need to establish and incorporate.
Sure. Well, I think that's a good suggestion, and I think it's still extremely important to have evidence-based recommendations, and including regulatory agencies. And I think if you make those recommendations, particularly evidence-based recommendations, and eventually, I think Big Pharma must listen to those recommendations and follow those guidelines.
Okay.
I really like this development, to develop guidelines for therapeutic trials internationally, but I see one danger, and this is complementary to what Lennart has said. If you do this in a big international group, it has to be done. Nothing against it. The problem may arise that it gets very complicated and difficult. What we need is what Lennart said. We need more simple trial designs so that they get funded, in particular by academic institutions. In Germany, for example, we get up to 2 or 3 million euro for the therapeutic trial. Up to 2 or 3 million euro. Otherwise, we can't do it. We cannot compete with medicine, internal medicine, and so on. They do it for this price. And therefore,
doesn't matter company. I mean, drug
no,
companies. Or
academically driven trials, financed by the German government, by the Deutsche Forschungsgemeinschaft, up to 2 to 3 million euro. And therefore, we should emphasize that we now try to make academic trials easier and more cost efficient, in my view, and not to do, let's say, complicated discussions. We have to end up with a pragmatic solution, in my view. This is my
very good. This question actually raises a very important point.